So this is a higher level development of the ideas really that we've already met in 2.7. And remember that the genetic code is essentially a blueprint, a recipe that makes proteins. And the way in which this works is that the gene is first transcribed into messenger RNA, and then that messenger RNA leaves the nucleus, goes out to the ribosome, and that's translated into a very, very specific sequence of amino acids. We also appreciate that this is really important because the protein that is produced from transcription and subsequent translation is the structural and the functional unit of the cell. So for any metabolic processes to take place, you need proteins. For any structural aspects of a new cell, you also require proteins. We also previously have come across this idea that the genetic code is obviously made of DNA, but the so-called previously considered junk DNA is no longer referred to as being junk. It's actually very, very important. And what we're going to focus on in 7.2 is really a digging down into the idea of the regulation of transcription and the regulation of the production of RNA. And this regulation is essential because you want to make proteins within a cell as and when is required. You don't want to produce them in a wasteful manner, and obviously you don't also want to produce them in deficit. So this idea of turning on transcription and turning off transcription and regulation of transcription is very much a focal part of 7.2. The syllabus statements are listed here. And again, what we need to appreciate for higher level is that it's all about regulation. We're going to think about regulation of the nucleosome. Remember that in eukaryotic DNA, we've got this protein, this histone protein associated with the DNA, and we can do methylation and acetylation of the DNA, which changes regulation patterns. We're also going to look at how certain proteins and certain non-coding regions of DNA can alter transcription. We're going to touch on epigenetics. We're going to look also about how the environment can directly affect gene expression. And we're going to also unpack more detail about non-coding DNA. This next image comes from BioNinja, and it's referring you to this idea that we've got a coding sequence, and the coding sequence is the DNA which ultimately is going to be transcribed into a single strand of messenger RNA. Then we've got various non-coding regions, and the promoter region and the terminator region are regions that we've touched on already. Remember that in transcription there are 3 stages. The first of these stages is initiation, and initiation requires RNA polymerase to be added to this so-called promoter region, but the promoter region itself is not then transcribed into a sequence of messenger RNA. The terminator sequence is similar. Remember also, this is just a reference to what we should already be aware of, that we've got 2 strands of DNA. We've got the sense strand and the antisense strand. Remember, it's the antisense strand, this one here, which is used as the template strand. So we're going to bring in nucleoside triphosphates into position using complementary base pairs. Remember that we've got uracil as opposed to thymine because we're producing messenger RNA. And also remember that this new sequence here is copied in this direction. So we're moving in the 5' to 3' direction. So it's this sense strand will be similar in terms of the base sequence, albeit replacing uracil with the sense strand with thymine. This also is the image that we came across earlier on, and we know that it's RNA polymerase is the enzyme that's responsible for bringing in these nucleoside trisephosphates into position and then subsequently having the energy, lowering the energy required to make this phosphodiester bond. So the direction is 5' to 3' in terms of the new strand of messenger RNA. And if we're talking about the DNA, it's obviously the opposite way around. So this next syllabus statement is asking us to really consider this idea of regulation. And what we're doing here is we're considering the promoter region, and the promoter is a non-coding region of DNA. And within this DNA promoting region, there are attachment points for various proteins and also RNA polymerase. Now, there are so-called transcriptional factors. Now, transcriptional factors are proteins, and if the proteins interact with either directly the promoter region or upstream, usually of the promoter region, that can have an impact on the gene expression. So if we scroll down here, we can see that we've got certain enhancer regions which are upstream of the promoter, or sometimes part of the promoter itself. If and only if there are certain proteins that are present and these activator proteins can bind to this particular region, what will happen is that will encourage the RNA polymerase to bind and do transcription at a faster rate. So enhancer regions are non-coding regions which speed up the rate of transcription. Also, by contrast, we've got regions of DNA, non-coding DNA, which are referred to as silencer regions. And again, if and only if we've got certain proteins present that can bind to the silencer region, these are known as repressor proteins, that what happens is we inhibit transcription and we stop the binding at the promoter region. This can be emphasized using the so-called lac operon gene. And within the Google Classroom, I've posted a video that unpacks this in quite a bit of detail. You don't need the detail that's there in that video for this part of the specification. It used to be in the old specification, but it's a useful example for us to consider. Now, this example of the lac operon is looking at non-coding regulatory regions in a prokaryote, and specifically it's E. coli. So what we can see in this diagram here is that we've got sequence here, which is the promoter sequence, and here we've got the RNA polymerase binding. And if we want to do transcription, then what we've got then is the direction of transcription here. But we've got in this region so-called operator regions, and the operator regions can either be enhancers or silencers, and depending on the proteins which are associated here, can change the rate of transcription. If we look at the next diagram, we can see this again in a little bit more detail. What we've got here is a repressor protein which is bound to this region, this operator region of the non-coding sequence of DNA, which means that the RNA polymerase, even if it binds to the promoter, can't do transcription. So it's blocking transcription. And here you can see it in context. And again, this underscores the importance of gene regulation. If the E. coli comes into contact with lactose sugar, then it needs to be able to break the bond between the monosaccharides to make glucose and galactose. And glucose and galactose can then be metabolized by the E. coli. So to do that, you need to have lactase, the enzyme. So what will happen is in the presence of lactose sugar, you've got the lactose sugar binding to this particular protein, and this particular protein now is going to change its shape, and as such cannot bind as it did to this repressor protein here, which means that transcription can take place. We can produce lactase, the enzyme to break down lactose sugar, and then encourage the breakdown of that sugar to be metabolized in respiration. In the absence of lactose, this doesn't bind, in which case the repressor protein will then associate and bind to this DNA operator region here, and it will prevent RNA transcription. So it means that you only produce lactase when you need to produce lactase. So this is just one example in a prokaryotic cell, but eukaryotes operate in rather the same way. And what we've got here is a bit of a summary, which is important. It's important to distinguish clearly in the language that you are using between the DNA sequence, or the control elements, as BioNinja calls them, and the protein that is going to block or activate those particular sites. So we've got the DNA sequences, which are enhancers. In order to speed up the rate of transcription, you need to have a binding protein that's present, and that will accelerate the rate at which RNA polymerase can then bind to the promoter region and do transcription. And then you've got the opposite here. You've got DNA regions which are called silencers, and only when repressor proteins are present, then you're going to block the transcription by binding of the repressor protein to the silencer region of the DNA. And the promoter, also important obviously for the binding of RNA polymerase to start this whole entire process. So all these examples here are non-coding regions of DNA. BioNinja goes into a lot more detail, and I've lifted this from BioNinja, and it talks about this idea of tissue-specific impacts of regulation. And what this means really is that the protein needs to be within the tissue in order for it to activate with the control elements, the silencers or the enhancers, to change the rate of transcription. And he's got an example that's outlined here. So this is from BioNinja. What we also need to recognize is that not only can regulation happen by the presence of proteins internally, but also we can change it due to external environmental impacts. So hydrangeas, these are flowers, and these will change the color of the petals depending on the pH of the soil that those flowers are placed in. And BioNinja has a nice diagram of that. The example that is in this PowerPoint here is that the Himalayan rabbit is going to produce different coloured fur depending on the temperature. So at the extremities, which are at the end of the rabbit's ears or the end of the rabbit's toes, we're going to produce black fur, whereas the body, which is much warmer, is going to produce white fur. We know from being exposed to the sunlight that we encourage the production of melanin. So the presence of UV light encourages the production of melanin within the pigment of the skin. And another interesting example is how certain species of fish, reptiles, and amphibians can change gender in response to particular social cues within the environment that they find themselves in. So psychological impacts as well. So this is just the example that shows the Himalayan rabbit. So you can see also that the same rabbit can change the coloration depending on the ambient conditions. And this next slide is interesting because it looks at gene expression in terms of the production in terms of embryonic development. And there are certain types of chemicals which can interact with the genes which are involved in development at different times. So they're called teratogenic agents sometimes, or morphogenic examples. A nice example that again goes beyond the syllabus, but it's interesting, is the use of an anti-pregnancy sickness drug called thalidomide. And thalidomide was prescribed as a wonderful drug within the 1970s, which stopped women having morning sickness. If this drug was taken at a particular time, however, during pregnancy, it could have very, very far-reaching impacts on the development of the limbs of babies. So thalidomide is an example of how an external chemical within the environment during embryology can have an impact. So this is the first video for 7.3, and we're going to look at more regulation in the next video, which is looking specifically at the nucleosome and methylation and acetylation patterns.